What factors associated with diabetes exacerbate atherosclerosis? And in particular, what are the relative roles of hyperglycemia, inflammation, and lipoproteins? And today I will talk about the lipoproteins. So the model we have used for a lot of these studies I will talk about today is a transgenic mouse model of type 1 diabetes accelerated atherosclerosis. And this model was developed many years ago now by Karin Renard when she was a postdoc in the lab. These transgenic animals express a viral protein called glycoprotein, or GP, under control of the insulin promoter on the pancreatic beta cells. This overexpression of or expression of GP doesn't cause a phenotype in the mice until you infect them with the virus, lymphocytic choriomeningitis virus, or LCMV. This generates an immune response in the animals, and we get the CD8 positive T cell mediated destruction of these GP expressing beta cells. This induces severe insulitis in the animals, and eventually within 7 to 10 days or so, a loss of insulin. Here you can see cross sections of an islet from a non-diabetic control animal where this brown reaction product indicates insulin. And here is an islet from a diabetic animal from of this model. And you can see very little brown insulin staining here. And actually we had to look around hard to find an islet that had some insulin staining. So insulitis and then loss of endogenous insulin production. Most of these animals require exogenous insulin to prevent severe weight loss and ketonuria. These animals are also crossed with the LDL receptor knockout mouse that I mentioned earlier to allow us to study atherosclerosis. So when fed a low-fat, semi-purified diet without added cholesterol, these diabetic mice are hyperglycemic, even though they do get some baseline insulin, and they have modestly increased plasma triglyceride levels in the form of triglyceride-rich lipoproteins, or TRLs. So what we have learned from this model so far is that mice with diabetes have an accelerated lesion initiation. This effect of diabetes can be prevented by an intense insulin treatment, which could be due to either glucose or TRL lowering. And that diabetes also promotes advanced lesions through a triglyceride or TRL dependent mechanism. So what are these TRLs? This is a simplified overview of metabolism of triglyceride-rich lipoproteins and their remnant particles in humans. And the TRLs are usually classified as chylomicrons. These are very large particles derived from the gut during and following a meal. These particles can range in size from 1200 to 80 nanometer in diameter. We also have the very low density lipoprotein that's produced by the liver in between meals. These are smaller particles, 80 to 35 nanometer in diameter. And then the remnants of these chylomicrons and VLDLs, which I will refer to as RLPs. These range in size from 80 to 25 nanometer or so, and they get progressively smaller as the VLDL and chylomicrons are hydrolyzed by lipoprotein lipase. And so then you get smaller and smaller RLPs. These RLPs are cleared through the liver, through LDL receptor family members and through syndecan, or they can accumulate in the artery wall. In humans, diabetes has been shown to increase production of VLDL, especially in cases where you have a triglyceride accumulation in the liver. And also diabetes prevents clearance of these RLPs. Another thing to point out, which will be important later on, is that these different lipoproteins carry different sets of apolipoproteins. So chylomicrons carry ApoB48, which is a truncated form of ApoB100 carried by VLDL and LDL. chylomicrons and VLDL and the remnants also carry ApoE. And ApoE has several different functions, but one is to mediate uptake, hepatic uptake and clearance of RLPs. And these TRLs also carry different ApoC, apolipoproteins, and ApoC3 will become important in a minute here. So in the lab, we have started to look more carefully at the lipoprotein and apolipoprotein abnormalities in diabetic mice using the type 1 diabetes model I described. And this work is being carried out by Masami, Shimizu, Albertine in the lab. And here, what we did was to separate plasma samples from non-diabetic and diabetic mice on FPLC, and then measured cholesterol in each fraction. And you can see that VLDL comes out first, and that diabetic animals have higher VLDL cholesterol than the non-diabetic controls. They also have higher levels of IDL, intermediate density lipoprotein, which is a type of RLPs, whereas there are no major changes in LDL and HDL in the diabetic animals. So when we look at the apolipoproteins in plasma by targeted mass spectrometry, we see consistent results with this. That is, there are no differences in the ApoA1 levels between diabetic and non-diabetic animals. And ApoA1 is the major structural protein of HDL, so that's consistent with the lack of difference in HDL cholesterol. There are also no differences in total ApoB or ApoB100, and that's because most of the ApoB is carried by LDL, where again, we have no major differences in diabetic animals. We detect several other apolipoproteins, and the main thing to point out here is that we see a significant increase in ApoC3 and ApoE in plasma from the diabetic animals. And the increase in ApoE is probably a reflection of reduced clearance of TRLs. Then Masami took the peak fraction here of the VLDL peak and looked more carefully at the apolipoprotein composition of VLDL and normalized that to the level of ApoB in these particles. And this will give you a rough estimate of the number of the amounts of these apolipoproteins or lipids per particle, because there is one ApoB molecule per VLDL particle. So you can see here that triglycerides and phospholipids were slightly elevated in the diabetic VLDL, but not significantly so, and that there were no differences in ApoC2 and ApoA5, which are activators of lipoprotein lipase. But the main difference we see is a very dramatic increase in ApoC3 carried by VLDL. ApoC3 is exchangeable between TRLs and HDL, and so we also isolated HDL by sequential density gradient ultracentrifugation, and again went back to do targeted mass spectrometry to look at the range of different apolipoproteins carried on HDL. And again, you can see that the picture looks pretty consistent. There is a significant increase in ApoC3 and ApoE carried by HDL from the diabetic animals. So this has made us very interested in the role of ApoC3 in cardiovascular disease associated with type 1 diabetes. And I've already told you that ApoC3 is an apolipoprotein that associates primarily with TRLs, their remnants, and HDL. Elegant genetic studies by Alan Schuldinger, Dan Rader, Rob Hegele, and Sek Katharisen has shown that ApoC3 null mutations or loss of function mutations confer TRL lowering and apparent CVD protection in humans. ApoC3 prevents clearance of TRLs and the remnants by at least two different mechanisms. It inhibits the action of lipoprotein lipase, so inhibits the hydrolysis of triglycerides in the TRLs and remnants. And it also prevents the hepatic uptake of these particles. An ApoC3 antisense oligonucleotide therapeutic called volanesorsen has recently been shown to reduce TRLs in humans, including in a small study in subjects with type 2 diabetes. But the role of ApoC3 in patients with type 1 diabetes, who often have normal plasma triglycerides, was unknown. So in order to ask questions about ApoC3 as a risk factor in type 1 diabetes, we teamed up with Janet Snellberg at the University of Colorado, Denver. She leads the coronary artery calcification in type 1 diabetes, or CACTI study there. This is a prospective study of people with type 1 diabetes, about 1,400 participants, 19 to 56 years of age, with a diabetes duration of 4 to 23 years. There was no known cardiovascular disease at baseline. So what we did was to first identify all coronary artery disease cases in total in the CACTI study. And that was defined as a myocardial infarction, bypass surgery, angioplasty, or CAD death based on medical records. And we found 47 CAD cases in CACTI so far when we initiated this study. We then analyzed serum ApoC3 levels by targeted mass spectrometry in baseline samples, so before the CAD events. And this is an example of a human-first approach. We set up a case cohort study where the case group contained all 47 CAD cases, and the cohort study was a randomly selected cohort consisting of 145 subjects with type 1 diabetes. So you can see here that the case group was worse off at baseline than the randomly selected cohort, in that they were slightly older, they had a longer duration of diabetes, they had elevated blood pressure, increased levels of total cholesterol, triglycerides, and LDL cholesterol, and their diabetes control was also a little bit worse than in the randomly selected cohort. I wanted to point out, though, that triglyceride levels were mainly in the normal range in both of these groups, with median levels of 75 and 99 mg per deciliter. So having measured ApoC3, we performed Cox proportional hazard models to look at ApoC3 as a CAD risk factor. And you can see here in the unadjusted analysis that for every 1 standard deviation increase in serum ApoC3 levels, there's about a 2-fold increased risk of having a later CAD event in these patients. Elevated triglycerides also increase the risk of developing a CAD event, but not as strongly as ApoC3. So this hazard ratio of about 2 remained significant when we adjusted the model for age, sex, diabetes duration, HbA1c, blood pressure, and smoking, and also when we adjusted the model for LDL cholesterol and HDL cholesterol. The significance was lost when we adjusted the model for log triglycerides, which was to be expected because ApoC3 acts by regulating triglyceride levels. So then we wanted to test mechanism in the mouse model, having established that ApoC3 is indeed a cardiovascular disease risk factor in type 1 diabetes. And these studies were carried out by Jenny Cantor. I'm sure you'll hear more from her in the future. She now has her own lab set up at the University of Washington. So she used the type 1 model that I described. Because these animals have a wide range of plasma triglycerides, what we did here was to divide the animals up into groups where the triglyceride levels were similar in the non-diabetic and in the diabetic animals within each bracket. And looking at these different brackets of triglyceride levels, when we measured plasma ApoC3 levels, the diabetic animals were always higher than the non-diabetic animals over this wide range of triglyceride levels. So why is that? Why is ApoC3 increased in diabetes in this model? Well, one possibility is, of course, that there isn't sufficient insulin or that the animals are hyperglycemic. So in order to dissect those two possibilities, we fed animals an SGLT2 inhibitor through the drinking water. And you can see here that blood glucose levels are, of course, high in the diabetic animals as compared to the non-diabetic controls. And when we gave them the SGLT2 inhibitor, the blood glucose levels were normalized. But plasma ApoC3 levels remained high in both of these groups, suggesting that hyperglycemia per se is not responsible for the elevated ApoC3. We then treated the animals with an intense insulin regimen. And as I said, these animals have some baseline insulin on board, but if we give them more frequent insulin injections, we can normalize their blood glucose levels fairly well as compared to these diabetic animals. And by doing so, we also normalized plasma ApoC3 levels. So this suggests that relative insulin deficiency rather than hyperglycemia per se is a cause of the increased ApoC3 in the mouse models. But is ApoC3 a causative factor in diabetes accelerated atherosclerosis? So the model we had so far is that in type 1 diabetes, probably because of reduced insulin levels, there's an increased production of ApoC3 from the liver. There are markedly elevated ApoC3 levels in circulation, primarily bound to TRLs. And this might lead to an increased accumulation of these remnant particles in lesions of atherosclerosis. But is ApoC3 driving this cycle? So in order to ask that question, we teamed up with Ionis Pharmaceuticals, who has generated the human antisense oligonucleotide and also has a mouse antisense for ApoC3. So we silenced ApoC3 in the liver during a 12-week study in the type 1 diabetes model. The animals were injected IP with either a control antisense or the ApoC3 antisense twice a week. So here you can see that ApoC3 antisense treatment was very efficient. We measured ApoC3 mRNA levels in the liver, and you can see animals that are diabetic have slightly elevated ApoC3 mRNA as compared to the non-diabetic animals. And ApoC3 mRNA levels in the liver were severely reduced in both of these groups by the antisense. Looking at plasma levels during this 12-week study, again, diabetic animals have much higher ApoC3 levels than the non-diabetic animals at all these time points. And when we silence hepatic ApoC3, the levels are basically gone. This was done using mass spectrometry and was later confirmed by an ELISA. ApoC3 normalized the TRL levels and ApoE in these diabetic animals. And you can see here is a lipoprotein profile again, where the diabetic animals are on top here, treated with a control antisense. And these are the diabetic animals treated with the ApoC3 antisense. So reduced triglyceride levels in the VLDL peak. It also reduced plasma ApoE levels in the diabetic animals without affecting plasma ApoB100. And I showed you earlier that diabetes also does not really alter levels of plasma ApoB100. Here you can see the drop in plasma triglyceride levels during the course of the 12-week study. So these are the diabetic animals treated with the control antisense, and here are the diabetic animals treated with the ApoC3 antisense. You can see that levels of triglycerides are normalized down to levels in the non-diabetic controls. These effects were not dependent on a blood glucose lowering, because in both of the diabetic groups treated with the control antisense or the ApoC3 antisense, blood glucose levels remained very high during this 12-week study. So what about atherosclerosis? We measured atherosclerosis at several different sites and by different methods. So this is an on-FOS preparation where the aorta is taken out, opened up longitudinally, and stained using a lipophilic stain called Sudan 4. And so these red patches you can see here are the lesions of atherosclerosis. And when we quantify that, diabetic animals have larger lesions than the non-diabetic controls, as we have seen many times before. And that was completely prevented when ApoC3 was silenced in the diabetic animals. We also investigated lesions in the brachiocephalic artery, which is a branch off of the aortic arch. And at this site, at the 12-week time point, the lesions were very early fatty streak type lesions. And most of the cells in these lesions were macrophages, as identified using a MAC2 antibody. And you can see the pattern is very similar to what we saw in the aorta, with diabetic animals having larger lesions than the non-diabetic controls, and that being completely prevented by silencing hepatic ApoC3. And of course, since these lesions are very early macrophage-rich lesions, when we quantify macrophage area in the brachiocephalic artery, we see a very similar pattern. Jenny also looked at whether ApoC3 accumulates in the artery wall by using immunohistochemistry. And the ApoC3 here is this brown reaction product in this very early lesion. You can see that ApoC3 has accumulated in the layer of the artery that faces the lumen, and that the diabetic animal here, given a control antisense, has much higher levels than the non-diabetic control. And this is quantified here. And of course, the ApoC3 antisense normalizes or prevents accumulation of ApoC3 in the artery. The same pattern was shown for ApoE, which is also present on TRLs and RLPs, and interestingly also for ApoB. And this is despite the fact that plasma ApoB levels were not changed by diabetes or the ApoC3 antisense. So these particles probably contain ApoC3, ApoE, and ApoB, and they accumulate in the diabetic state. Jenny also set up a study to look at more advanced lesions. And the way we did that was to first feed animals a high-fat diet for 12 weeks to allow them to develop advanced lesions of atherosclerosis. They were then fed a chow diet to normalize or reset plasma lipids for 2 weeks, and diabetes was induced by LCMV. And then we did a 4-week diabetes study where animals were treated either with a control antisense or the ApoC3 antisense. And you can see in this case, the lesions are a lot more advanced with large necrotic cores and cholesterol clefts. And we didn't really expect a difference in lesion size in this study because the animals had large pre-existing lesions before we induced diabetes. And that's indeed what we saw. And also ApoC3 antisense did not affect the size of these lesions. But interestingly, it reduced the necrotic core area. And this is a sign that ApoC3 antisense prevents progression of these lesions to more advanced stages. And ApoC3 antisense also reduced the ApoC3 accumulation in these arteries. We've recently started to look more closely at the co-localization of ApoC3 with ApoB and macrophages in lesions of diabetic mice. And here is an example of a lesion that was co-stained for ApoC3, which is this green reaction product here, ApoB, which is this purple reaction product. And you can see that there is a close overlap between these two stains. And then macrophages shown in red here. So in these lesions, ApoC3 and ApoB co-localize just below where the macrophages are accumulating in these lesions. And this concept is supported by the following experiment in which we harvested peritoneal macrophages from the non-diabetic and diabetic animals and animals treated with the antisense. And so when you do that, we see that macrophages from diabetic mice in the peritoneal cavity contain more cholesterol esters, so they're more lipid loaded than the non-diabetic macrophages. And that again was normalized by ApoC3. That reflected the interstitial fluid cholesterol levels here, and also the interstitial fluid ApoC3. So probably in these interstitial spaces and maybe also in the lesion, cholesterol levels and ApoC3 levels can affect the cholesterol loading status of the macrophage. So the model so far then is that we believe that ApoC3 has a heightened importance as a cardiovascular disease risk factor in the setting of diabetes because it's regulated by insulin. And so in type 1 diabetes, relative insulin deficiency or perhaps insulin resistance leads to an increased production of ApoC3 from the liver, increased plasma levels of ApoC3, and these are primarily found in the TRLs. This causes a prolonged circulation time of these TRLs because they're not cleared as efficiently as they should be, not hydrolyzed. And that we believe results in an increased accumulation of TRLs or the remnants in the artery wall. These remnants are then taken up by macrophages. We get macrophage foam cells and increased atherosclerosis and an increased risk for coronary artery disease. So why do these particles accumulate in the artery wall in the setting of diabetes? Is it just that they circulate it for longer and is not hydrolyzed to the same extent as in the non-diabetic setting? Or could there be local changes in the artery wall that makes these particles be trapped more in the diabetic state? So it's well known that ApoB, which is present on these particles, has positively charged sites, and there's a particular site called site B that's known to bind to negatively charged glycosaminoglycan chains in different proteoglycans. And these are part of the extracellular matrix in the lesion and in many other tissues. So we were particularly interested in Versican because we had some preliminary findings that Versican mRNA was upregulated in the setting of diabetes. And so in order to investigate the role of Versican, we collaborated with Chuck Freevert and Tom White in Seattle to use mice with myeloid cell Versican deficiency that they had generated. And we set this study up similar to what I showed you before to look at more advanced lesions. So we fat-fed the animals for 12 weeks. Then we gave them bone marrow transplants from wild-type animals or animals with a myeloid cell restricted Versican deficiency. We let them recover for 5 weeks, and then we induced diabetes for 4 weeks. And so at the end of this study, we indeed saw that macrophages from diabetic from non-diabetic animals, and the knockouts were knockouts. Knocking out Versican in myeloid cell did not affect blood glucose levels, plasma cholesterol levels, or plasma. Sorry, is there a problem with the connection?
Having a little bit of a problem, but mostly can hear you.
Okay, great. So when we did that, we looked at the sinus lesions again, and there was no difference in lesion size. But by knocking out Versican in the myeloid cell compartment, we saw a reduced presence of Versican in these lesions. So some of the Versican in the lesions is indeed coming from the macrophages or myeloid cells. Knocking out myeloid Versican did not affect the elevated plasma ApoC3 levels in the diabetic animals, but it did reduce accumulation of ApoC3 in these sinus lesions. And you can see that measured two different ways here. So this suggests that it's not only an increased level of these TRLs in plasma and reduced clearance of these particles that's important in diabetes, but also changes in the extracellular matrix in the lesion could perhaps lead to increased trapping of these TRLs or RLPs in the lesion. So the last bit of the study I wanted to tell you about is just now in press. And in order to look at in people with diabetes, type 1 diabetes, whether insulin resistance could contribute to the increased ApoC3 levels, we went back to the cacti study and went back to this randomly selected cohort. In this study, we also stratified the study based on sex. So you can see men here and women in this column. And the men had higher blood pressure than the women. They also had a significantly higher waist circumference and higher ApoB levels than the women in this cohort. And the women had higher levels of HDL cholesterol. Then we measured insulin sensitivity through a formula that I will describe in a minute. And insulin sensitivity was lower in men than in women, suggesting that the men were more insulin resistant than the women. And this formula has been validated to be very similar to the HOMA-IR method. It's based on a formula calculated based on waist circumference, insulin daily dose, adiponectin, triglycerides, and diastolic blood pressure. And so when we used association between serum ApoC3 and insulin sensitivity, we found that only in males was increased ApoC3 associated with insulin sensitivity, so negatively associated with insulin sensitivity. So it's actually positively associated with insulin resistant in men with type 1 diabetes. And so was ApoB and non-HDL cholesterol. We also looked at the association with coronary artery calcium in these same subjects. And since we had coronary artery calcium on all of these, and we found, which is consistent with our previous findings, that ApoC3 is a predictor of coronary artery events, that ApoC3 was positively correlated with coronary artery calcium, and that was independent on gender in this cohort, and so was ApoB and non-HDL cholesterol. ApoC3 bound to HDL was not significantly correlated, but definitely showed the same trend. So building on this model then, in type 1 diabetes, we believe that the increased ApoC3 could be due to either relative insulin deficiency or to increased insulin resistance, at least in men. So let me summarize this. We have found that elevated ApoC3 increases the risk of having a coronary artery disease event in people with type 1 diabetes. The elevated ApoC3 is due, at least in part, to relative insulin deficiency and or to insulin resistance. So together, we think these studies point to a heightened importance of ApoC3 in cardiovascular disease associated with type 1 diabetes. But many important questions remain, of course. For example, what is the mechanism? How does insulin action suppress ApoC3 in diabetes? Are similar mechanisms at play in type 2 diabetes? Very important question. And perhaps most important of all, would ApoC3 inhibition prevent the residual cardiovascular disease risk in humans with diabetes? So I have many people to thank, of course, and first, the incredible team at the UW Diabetes Institute and members of my group here. And I've tried to point them out as I went by, but Jenny Cantor did almost all of the ApoC3 work I talked about today. Masami Shimizu Albergine is also working on a similar project in the lab. Farah Kramer is an expert in taking care of our type 1 diabetic animals. And we have many important collaborators at the UW. Jay Heinecke, Thomas Weiser, and Bohai Shao are experts in mass spectrometry, and they are responsible for all the nice mass spec data I showed during the talk. And Bohai Shao is shown here. We also collaborate with Carla Greenbaum, Tom White, and Charles Frewer were collaborators on the Versican part of this story. Of course, I'd like to thank Janet Snellbergen for giving us access to the important cacti samples, Ira Goldberg and Ed Fisher at NYU for collaborating. Of course, Ionis for providing the ApoC3 antisense and our funding sources. And importantly, also the Diabetes Research Center, of course, at the University of Washington, who is providing important core services for this type of research. So with that, I'd like to thank you for your attention, and I'd be very happy to take any questions you might have.
Thank you, Karin. That was wonderful. We do have a number of questions from the audience. First, Jane Roish. I'm going to allow you to talk. Jane, if you want to unmute yourself, you can ask a question.
Yeah, I think I am unmuted. First, Karin, thank you. That was really beautiful. And both your generation of these excellent models and the detail with which you've unraveled this story is fascinating over the last 20 years. So I was really intrigued by the fact that the lipid core decreased with the ApoC antisense. And so that in that period of time, that seemed like an active process. Do you think there's also lipid unloading going on?
We think that in that model, it's mainly prevention of the effect of diabetes to lead to progression of these more advanced lesions. So it's probably more an inhibition or a prevention of expansion of these necrotic cores rather than cholesterol removal. But we haven't fully investigated that possibility yet.
Thanks.
Okay, our next question is from John Stafford. John, I'm going to allow you to talk. You can go ahead and unmute yourself.
Yes, thank you for a great talk. There's evidence in people with diabetes, type 2 diabetes, that they have this chronic sort of smoldering damage to the heart that may be at least in part due to the small arteries. And this also may contribute to stroke in type 2 diabetics. Do you have big picture thoughts on how we can better study that using mouse models?
That's an interesting.
Those small arteries
in the heart. Well, yeah, that's
in brain.
In the brain. That's an interesting question. We haven't really done that ourselves. I think that there are more and more sophisticated imaging methods to look at smaller vessels now, but I'm not exactly sure if we are able to already do that in the small vessels in the brain. So there might be other people in the audience who have better ideas here than I do.
Thank you.
Okay, next question from Alessandro Vito. You can go ahead and unmute yourself.
Hi, yeah.
Go ahead and unmute yourself, Alessandro.
Okay. I should be unmuted. Am I unmuted?
Yep.
All right, awesome. Thank you. Okay, great presentation. Thank you so much. So I was wondering that another major risk factor for atherosclerosis and cardiovascular diseases in general is just biological aging, just getting older. So do you know if there's any data on changes in on the levels of ApoC3 and its dynamics with age in either mice or humans?
Oh, yeah, that's an interesting question. Well, I think we could look at that. And of course, in aging, there could be increased insulin resistance or reduced insulin sensitivity, which could potentially increase ApoC3 levels. I am not exactly aware of any study where they have followed mice, aging mice or humans, but there might be such studies. I think that's a very important question because for sure, aging is a very important cardiovascular disease risk factor.
Thank you so much.
Okay, next question, Alan Atzi. You can go ahead and unmute yourself, Alan.
That was a lovely talk, Karin. So as you know, hyperinsulinemia has been linked to atherosclerosis, but your studies show that insulin deficiency is associated with atherosclerosis. So that's a bit of a paradox. How do you explain that?
Well, I think maybe hyperinsulinemia could be a result of or coexist with insulin resistance, right? So in the liver, even if you have hyperinsulinemia, you might have hepatic insulin resistance that could potentially lead to increased ApoC3 levels. That's how I think about it. I think insulin in general has not really been shown to be directly atherogenic. So the association between cardiovascular disease risk and hyperinsulinemia might be more of an indirect effect.
Do you know if, there have been some models where the insulin receptor has been knocked out in endothelial cells. Do you know if in those models there's a difference in atherosclerosis susceptibility?
Yes, in those models, it varies depending on where you knock the insulin receptor out. If you knock it out in endothelial cells, George King has shown that you get increased atherosclerosis. No, reduced atherosclerosis, I think. I might have this wrong, but if you knock it out in myeloid cells, different groups have found different effects. And then, of course, there are other studies where it's been knocked out in the liver, for example. So it varies depending on where you knock the insulin receptor out, if you have a pro-atherogenic or an anti-atherogenic effect.
Thank you.
Next question, Bob Eckel. Go ahead and unmute yourself, Bob.
Okay, we're good now. Thanks, Rebecca. Hey, Karin, nice work. And, I'm a little bit familiar with some of it anyway. The question I have in terms of mechanisms by which insulin resistance or insulin deficiency may give rises to increases in ApoC3 containing lipoproteins. So the question more directly, doesn't insulin affect the transcription of the ApoC3 gene? I mean, aren't there enhancer elements in the proximity of that gene that modifies its transcription? And is it possible that that is a major reason why insulin deficiency and insulin resistance modifies C3 containing lipoproteins?
Yeah, and in fact, we did look at that a little bit. So it's been described that FOXO1 regulates ApoC3 transcription in the liver. There are also studies showing that hyperglycemia can upregulate ApoC3 in the liver. We didn't see a clear role for the FOXO transcription factors, and in collaboration with scientists at Columbia University, Rebecca Hausler there, we knocked out all the FOXOs in the liver and didn't really see differences in ApoC3 mRNA levels in those animals. So it could be still that insulin regulates transcription or suppresses transcription of ApoC3. We just don't quite understand the mechanism yet. And that's an area we're very interested in pursuing.
So your mRNA data were in the liver only. Did you look at the gut also?
No, we haven't looked. Well, we have looked a little bit at the gut, and the antisense oligonucleotide that we have used primarily targets the liver, and we don't really see changes in the intestinal expression of ApoC3. So we think that the liver is the most important site of ApoC3 production, responsible for the changes we see in this model.
So that was the GalNAc construct, right, that you used?
This was actually predating the GalNAc construct, but it still targets mainly the liver, because probably delivery is better to the liver than to the intestine.
Okay, thank you.
Thank you. Next question, Michael Brownlee. Go ahead and unmute yourself.
He doesn't usually have a working microphone, so you probably have to read his question.
Thank you. I will do that. He asks, or he says, very exciting work. Ira Tabas has stressed that the importance of reduced specialized pro-resolving mediators, or SPMs, and increased leukotrienes as promoters of instability of atherosclerotic plaques. Do you have any data from your models relevant to this?
No, unfortunately, we don't have any data related to that yet. I think that's a very important question, a very interesting question, but unfortunately not yet.
Okay. Next question, I think Hunter Vessels. You can go ahead and talk. Unmute yourself, please.
Great, thank you. I was curious, I'm trying to study some of the interface between diabetic complications and genetics. And so my question was whether you had looked in human genomic studies. Is there a variant in ApoC3 that infers cardiovascular risk independent of diabetes?
Well, loss of function mutations in ApoC3 confer cardioprotection in people without diabetes, and I assume also in people with diabetes, but these mutations are pretty rare. So we don't have a sufficient number of subjects with these loss of function mutations to really stratify them into people with and without diabetes. But in general, loss of function of ApoC3 is cardioprotective in humans.
Thank you.
Okay, we still have several questions. Next, Ana Patricia Valencia, go ahead and unmute yourself.
Hi, Karin. Thank you so much. This was fantastic work. So long term, do you think that inhibiting ApoC3 could potentially lead to increased fat deposition in the liver and consequently insulin resistance? Kind of like if this was became a drug, like long term, do you think it would actually lead to problems, the same problem eventually?
Right. In this mouse model, we didn't see any changes in liver fat up to 12 weeks of treatment with the antisense. That is, of course, a little bit difficult to interpolate to human studies, but so far we haven't seen any signs of hepatic lipid accumulation in inhibiting ApoC3.
Okay, great. Thank you.
Okay, the next question is from Rebecca Schneider-Aguirre, who does not have a working microphone, so I'm going to go ahead and read it. She says, thank you for your presentation. I'm trying to extrapolate in my mind relative insulin deficiency and ApoC3 levels to humans and how this would help reduce CVD. You showed how blood glucose control with SGLT2 inhibitors did not change ApoC levels, but that lower blood glucose with insulin did. However, in type 1 diabetes, relative insulin deficiency is usually reflected by having poor control. Do you have any thoughts as to whether there is a certain amount of insulin that changes ApoC3 levels independent of blood glucose control? That is, could a patient have hypoglycemia as long as they run a certain amount of insulin?
Yes, so in people with type 1 diabetes, of course, you don't have such severe insulin deficiency as we do in these animals, although perhaps at times there would be relative insulin deficiency. It's possible that in people with type 1 diabetes, hepatic insulin resistance could be more important in driving the elevated ApoC3. And it's important to point out that ApoC3 is not universally increased in people with type 1 diabetes as compared to people without diabetes. And so we think that it's in cases where you have poor control perhaps, and maybe some features of insulin resistance or the metabolic syndrome. Those are the subjects with type 1 diabetes who are most likely to have elevated plasma ApoC3 levels, and who are also most likely to go on to have a coronary artery disease event.
Okay, great. Next question from Josh Castillo. Go ahead and unmute yourself, Josh.
Hi, Karin. Fascinating talk. I was just curious if you saw a decrease in your in the presence of macrophages in your aortic lesions with your Versican knockout model.
We did not see that, and of course, measuring the amount of macrophages at the static time point is a little bit difficult in these models. It might not reflect the rate of exchange of these cells in the lesion, but yeah. So just looking at one time point like that, we did not see a drop in macrophages.
And the next question from Al Applin. I'll go ahead and unmute yourself.
Hi, Karin. That was a great talk. I have a real quick question. If you know anything, or do we know anything about APO3C in other models, other animal models of athero?
Jenny Cantor is working on a type 2 diabetes model that also has kidney disease, and it's definitely elevated in that model of type 2 diabetes and kidney disease. That's one model I can think of now. There are also transgenic animals that are available. There's a transgenic mouse that has elevated atherosclerosis.
Okay, last question from Sujal Lee. You want to go ahead and unmute yourself?
Oh, hi. Yeah, a very nice talk, Karin. Thank you. Thank you very much. And my question is like, since in your the background graph, you show like the ApoC3 could work between the on liver cells and also then the lesion macrophages. And I wonder, have you ever tested to see if it has any effects on adipocytes, which is also another major player on diabetes and insulin resistance?
Mm-hmm. We haven't directly tested that yet, but in the type 1 diabetic animals, of course, they have very little adipose tissue, but we haven't seen a difference in the amount of adipose tissue, for example, in these animals. That's really all I can say about adipose tissue effects in the type 1 diabetes model at present.
Okay, thank you. Yeah, maybe when you test the type 2 diabetes model, we can see if, yeah.
Yes.
Thank you.
Thank you, everybody, for such a fantastic discussion. And thank you again, Karin, for a great talk and engendering such a great discussion. Just wanted to remind everybody again, Sean put information in the chat for trainees who might want to take more of an opportunity to talk to Karin more about her work on Friday this week. She's going to host a roundtable. Please contact Linda if you are interested in doing that. And then our next event is our first panel discussion, as Sean mentioned at the beginning. This will be on Wednesday, December 9th, and it will be a 90-minute session. Here are our speakers. We're very excited about this session. It's a little bit of something for everybody, and so we really hope that you will be able to join us for that session on the 9th of December. Many thanks once again, everybody, and have a great rest of your day.
Yes, thank you, everyone.
The recording has stopped.